Hello there, ladies and gentlemen, and everything in between. My name is Ryan McDowell. Tonight, we're going to have a brief and detailed presentation on the ACE2 transmembrane protein, considered a zinc metallic protein because it does have a molecule of zinc in it. Fun fact. The ACE2 protein, now the ACE standing for angiotensin converting enzyme 2, is a very integral part of a very complicated metabolic mechanism known as RAS, or the renin-angiotensin aldosterone system. That's something that we'll get into a little bit later, but it's important to note that decoding the mysteries of these cells can unlock a key to very, very powerful therapeutics. So without understanding all of the cell mechanisms and everything we've been studying, we wouldn't ever know how to apply it and how to get ourselves out of some sticky situations. Now, to remind you, bring you back a little bit, a little throwback to exactly what a transmembrane protein is here. You'll notice transmembrane protein is the one that goes all the way through the membrane of the cell here. And if you'd remember, it's a semi-permeable membrane comprised of the phospholipids that stick end to end, and it doesn't allow everything through. So remember, it's pretty selective. And so, especially when it comes to foreign particles and pathogens, and we want that, that's a good thing. But sometimes some things can trick our cells into gaining entry by some of those transmembrane proteins, and we are going to touch on that later. But I wanted everyone to remember here exactly what the membrane looked like and what it looks like when a protein permeates that membrane entirely. You've got a part sticking out of the cell, and you've got a part that goes into the cell, and the pore that allows access through that membrane and to the cell. Now, here we are going to touch on the metabolic pathway that I was referring to before. Now, that enzyme, ACE2, that we just referred to as part of this complex metabolic pathway. The ACE2 protein here, you'll notice, is just down here, angiotensin 2, if you'll see it right about here. And so we'll touch on that, but we'll start. I don't want you to get too overwhelmed here about the metabolic system. It's fairly simple. We can follow it together here. We'll start at the liver and angiotensin here. Now, angiotensin is acted on by something called renin that's released from the kidneys. And so this is all triggered by a drop in blood pressure or a drop in fluid volume. I don't want to talk about the drop in fluid volume here because that's bad news, but a drop in blood pressure can happen fairly regularly. And so when this happens, renin is released from the kidneys, and then angiotensin is released from the liver. Now, renin acts on angiotensin to form angiotensin 1, which is its first conversion step here. Now, ACE, oh, well, that looks familiar. ACE is the angiotensin converting enzyme, but that's the first version of this. And this is released from the lungs, which changes our angiotensin 1 to angiotensin 2. Nice. All right, and so once we have that action here, this angiotensin 2 acts on the adrenal glands to stimulate the release of aldosterone, which helps us absorb some more water and salt in the kidneys, and that changes our blood pressure and volume. And so you can see here it's a big closed loop system where angiotensin 1 and angiotensin 2 act as opposing forces to either constrict or dilate the blood vessels to allow flow for the kidneys. And so I know that might have been a mouthful or a brainful or an earful or whichever one you'd like to choose, but that's the metabolic pathway that we have to think about and all of the different places that that pathway exists and the receptor that could be there in all of those different areas of the body. Now, one of the things that I find extremely fascinating is that conversion that takes place in between angiotensin 1 and 2 is very similar to this conversion here that takes place. And this is the conversion between angiotensin 1 and angiotensin 1,7. And these are the things that either vasoconstrict or vasodilate. And I just really thought it was fascinating to see these structures so similar. And in the body, they're just really, really big difference. They do completely opposite things. So that's just really cool. And you might be asking yourself, hey, what's exactly does this have to do with anything? Well, I was alluding to it a little bit before, but this happens to have a lot to do with what's going on in the world right now. And if you haven't guessed it, it's the SARS CoV-2 virus that actually happens to gain entry to our cells through the ACE2 enzyme. And so we'll just do a brief overview on how a virus works. We've got a few steps on how it gains entry and then gets basically created and reproduced and released by our cells. And so it goes a little something like entry and attachment, which in this case, in this case of a virus that we might know, it gains entry through that ACE2 protein. But generally speaking, it's entry and attachment. And then you go through a little step called translation, where usually it takes some genetic material and gets it in the right format, essentially. From there, we replicate it, which is the copying and duplicate, making a bunch of different copies. And then from there, we go to assembly. Assembly is where it's put together into what's called a virion. And that's the viral particle that's eventually released into the cell. And that's once it's released, that's bad because, of course, it can infect either other cells that are nearby or other hosts, which is optimally what it wants to do, and which is what the SARS-CoV-2 virus is really good at doing. And so we started from the bottom, now we hear type thing when it comes to information on SARS-CoV-2 virus. And so it, might have almost seemed a daunting task at the beginning, but now we have so much information that we're almost overflowing with it. And so one thing we do now know, and that we knew before with the SARS-CoV-1 virus, is that it gains entry through the ACE2 protein through this very specific structure on the surface of it called the spike protein, right? You may have heard a thing or two about the spike protein, but there's one very specific region on this spike protein called the receptor binding domain. And that is that special region that I implore you to do further research on because it's really incredible, is what interacts specifically with the ACE2 gene and essentially tricks, pardon me, transmembrane protein. It tricks that transmembrane protein into allowing entry to this big old cell here. Oh, goodness. And this cell, once it gets inside, it really wreaks havoc, And so you can see here the steps, it's recognize each other. We've got a little bit of attachment here. And then once we have binding, we've got a few cleavage events where basically some things are broken off the membrane here and off the virus. And those start a chain reaction of events that allow entry into the cell, eventual endocytosis. And from there, it's bad news bears because SARS CoV-2 and CoV-1, as well as MERS, have RNA ready DNA, well, RNA ready RNA, sorry. I'm sorry, ribosome ready RNA. That's the RNA that's contained within it is ready for the ribosome. And so it can go straight to the ribosome and begin copying. And so this lovely slide here that I transitioned to, just without you even noticing, maybe, hopefully, is all of the potential therapeutics that we have for the SARS-CoV-2. And I don't want you to get overwhelmed by this because really we're just looking at this little guy right here, ACE2. And maybe you've heard of chloroquine. That's not something we will talk about too much, but I wanted you to just respect all of the lovely science that goes into this and the fact that we know so much now. And without this knowledge, we would, not have even close to the therapeutic potentials that we have currently. And this is all to help us combat this here. This is a slide of all of the emerging pathogens and infectious diseases that are in the world right now. And so if you thought the last slide was daunting, I mean, look at this. These are all the things that could potentially infect us. And so I think the real issue here is that we need to constantly pursue science. We need to be ever vigilant. And we need to make sure that we don't neglect these issues that have been brought to light locally and recently and globally, but we can't let them fall by the wayside again like they may have after the pandemic of the early 1900s. I feel like there was a lot of lapse in concentration and really public knowledge of viruses and the things that go into public health. And so I hope that we can continue our constant pursuit of knowledge here and remember that we can't stop ever, because we might just lose this race if any one of these things gets the upper hand. To review, we've reviewed the ACE2 SARS-CoV relationship, transmembrane proteins, and enzyme activated pathways, specifically as it relates to the ACE2 and the renin-angiotensin-aldosterone system. We took a brief visit into the world of the viral life cycle, as well as touched on how ACE2 and SARS-CoV-2 are interact with each other in a very specific way to gain viral entry. We touched on, too, those therapeutic potentials of ACE2 inhibitors, and other mentionable things, as well as the real issue here. If we aren't able to constantly pursue these scientific endeavors, then we might just fall behind and get caught by one of these viruses. Thanks for your time.